Hello, everyone. Our lesson for today is about intracellular membrane traffic of proteins and other molecules. In this lesson, you'll be able to learn about the molecular mechanisms of how proteins and other molecules are secreted, taken up, and retrieved within the cell through vesicular transport. An overview of this type of transport will be given in the first part. We followed by a discussion on its molecular mechanisms. Transport to and from specific cellular compartments, like the ER and Golgi apparatus, will also be elaborated. At the end of this lesson, you will be able to summarize the general flow of the vesicular transport pathways, describe the molecular mechanisms of membrane transport, and discuss the mechanisms of secretory and endocytic pathways. Before we go to our discussion proper, let me ask you this question. Have you wondered how proteins and other molecules travel from one cellular compartment to the others within a cell? I hope you will be able to answer that one after we discuss this lesson. In our previous lesson, we talked about the three types of protein transport. So it could be gated transport, transmembrane transport, and the vesicular transport. We've also discussed the other two, the gated transport and transmembrane transport, in our previous lesson. This lesson is solely dedicated to our discussion on the vesicular transport processes. We know that every cell must eat, communicate with the world around it, and quickly respond to changes in its environment. To help accomplish these tasks, cells continually adjust the composition of their plasma membrane and internal compartments in rapid response to need. They use an internal membrane system to add and remove cell surface proteins such as receptors, ion channels, and transporters. Through the process of exocytosis, the secretory pathway delivers newly synthesized proteins, carbohydrates, and lipids either to the plasma membrane or the extracellular space or out of the cell. In contrast, cells remove plasma membrane compartments through endocytosis and deliver them to internal compartments called the endosomes, from where they can be recycled to the same or different regions of the plasma membrane, okay, or be delivered to lysosomes for degradation. So cells can also use endocytosis to capture important nutrients such as vitamins, cholesterol, and iron. So basically, as what we have discussed, no, I think in our module number 2, exocytosis is when the cell secrete proteins or other substances outside of the cell, and endocytosis is when they take up these nutrients or other substances. Now, the only difference with this lesson is that we will be discussing these different processes at the molecular level. Okay. We know that the interior space of the endoplasmic reticulum, called the lumen, is also continuous with the perinuclear space, no? Because the outer membrane of the nuclear membrane is continuous, no, with the endoplasmic reticulum membrane. So the interior space, called the lumen, of each membrane-enclosed compartment along the secretory and endocytic pathways is equivalent to the lumen of most membrane-enclosed compartment of the cell exterior. So they are almost the same. So you have here the lumen of ER, which is the same with the lumen of Golgi apparatus and other organelles. This is also the same with the lysosomes and stuff. Proteins can travel in space without crossing a membrane. So they don't cross the membrane anymore because they have these transport containers. Okay? So one container will just bud off from one compartment and will just fuse to another compartment. So these containers are formed from the donor compartment and are either small spherical vesicles, larger irregular vesicles, or tubules. All these forms of containers are called transport, vesicles. So within eukaryotic cell, the transport vesicles continually they bud off from one compartment to the other. So say for example, you're from ER to the Golgi, and through these transport vesicles, you can find the proteins, other materials, the lipids, and some other soluble luminal molecules, and we call those materials as the cargo. So it is the traffic here is very organized, no? So the flow from the ER to outside and from the outside to the ER is very organized. Okay, so retrieval pathways, the process of going back, to the ER is important because part of it has already been lost, when the transport vesicle buds off. That's why another vesicle must also fuse with it in order for the ER to still be intact. Otherwise, if all the cargoes will be taking parts of the ER in the process and no retrieval, then ER will be it will be off. It will be finished. Okay. So each transport vesicle, they must bud from the that would bud from the ER must be very selective as well. No, so they need to take up only the appropriate molecule and also fuse with appropriate target membrane. Otherwise, it will be a waste of energy and waste of the materials if they do that. Okay, a vesicle carrying from the ER to the cargo apparatus, for example, must exclude the proteins coming from the ER and must take only the proteins or other materials that are needed not to their destination. So directed and selective transport of particular membrane components from one membrane enclosed compartment to another in a eukaryotic cell maintains the differences between those compartments. So we will now be talking here about the molecular mechanisms of membrane transport. Transport vesicles, which can be spherical, tubular, irregularly shaped, bud from specialized coated regions of the donor membrane. So these different vesicles are coated as well. Okay, so the assembly of the coat helps to collect specific membrane and soluble cargo molecules for transport and to drive the formation of the vesicle. What it means is that one type of coat for a particular cargo specifies that it is a secretory. Another type, it is endocytic. Another type is for the retrieval. So what are these different types of coats? So there are various types of coated vesicles. The best characterized are the clathrine coated vesicles, which mediate the transport from the plasma membrane and the trans-Golgi network. Okay? And the COP1 and the COP2 coated vesicles that mediate transport between the Golgi cisternae and between the ER and the Golgi apparatus respectively. So again, the clathrine coated vesicles mediate the transport from the plasma membrane, okay, the plasma membrane here, and the trans-Golgi. Okay, so we know that the Golgi apparatus is of three components, or three phases. You have here the cis Golgi, which is the one that is facing the ER, the medial, and the trans Golgi, the one that's facing the outer membrane. Okay, so the transport of substances from that is taken up by the cell through the cell membrane and into the trans-Golgi network, the TGN, okay, is through clathrine coated vesicles. On the other hand, COP1 and COP2 vesicles are the ones that mediate between Golgi cisternae, the cis, and between the ER and the Golgi apparatus respectively. Okay, so you have here COP1 vesicles, okay, and you have here the COP2 vesicles. Okay, COP2 from the ER to the Golgi. COP1 is needed for the transport from the Golgi back to the ER. So codes have common two-layer structure. An inner layer formed of an adapter proteins linked to the outer layer or cage of the to the vesicle membrane, and also traps specific cargo molecules for packaging into the vesicle. The coat is shed before the vesicles will fuse with its appropriate target membrane. So local synthesis of specific phosphoinositides creates binding sites that will trigger the clathrine coat assembly and vesicle budding. So this is for the endocytic pathway, okay? Also, the monomeric GT phases here help regulate various steps in vesicle transport, including both vesicle budding and docking. So you have here the fusion, the docking, and the docking, and this is the fusion. Okay, so the coat recruitment GT phases, okay, including SAR1 and ARF proteins, regulate coat assembly and disassembly. A large family of RAB proteins functions as vesicle targeting GT phases. RAB proteins are recruited to both, forming transport vesicles and target membranes. So the assembly and disassembly of RAB proteins and their effectors, specialized membrane domains, are dynamically controlled by GTP binding, and hydrolysis. Okay, so active RAB proteins recruit RAB effectors such as motor proteins, which transport vesicles along actin filaments or microtubules, and filamentous tethering proteins that help ensure that the vesicles deliver their contents only to appropriate target membrane. Complementary V snare, okay, and T snare are on the transport vesicles and T-snare proteins on the target membrane. They form the stable trans-snare complex, which form the two membranes in close opposition so that their lipid bilayer can fuse. Just like that. So, how is the transport from the ER through the Golgi apparatus? We know that this is already transmediated by COP2 coated transport vesicles. They pinch off from the ER, okay, and the vesicles shed their coat and fused with vesicular tubular clusters. So in animal cells, the clusters then move on to microtubule tracts to the Golgi apparatus, where they fuse to form the cis-Golgi network. Any resident ER proteins that have already escaped or have included in the vesicle will be delivered back to the RAF ER via the retrograde transport in COP1 coated vesicle. If there are something that should not be included here and needs to be returned, then they could be returned into the RAF ER through the COP1 coated vesicles. The Golgi apparatus, unlike the ER, contains many sugar nucleotides. So this is during the transport from the ER to the Golgi apparatus. So these sugar nucleotides, which glycosyl transferase enzymes use to glycosylate lipid and protein molecules as they pass through the Golgi apparatus. So say for example, proteins are now delivered from the RAF ER to the Golgi apparatus. So there are enzymes there called glycosyl transferase, okay? And in there, sugar nucleotides will be added. The mannoses on the N-linked oligosaccharides are added to the proteins in the ER and are often initially removed. So because in the ER, during protein folding, N-linked oligosaccharides are already added. Part of the protein modification process that occurs in the ER is the N-linked glycosylation. Now, as it is transported through the Golgi apparatus, glycosyl transferases will initially remove some of the sugar, like the mannoses, and will add further sugar nucleotides into the protein. So moreover, the Golgi apparatus is the site where O-linked glycosylation also occur, and where glycosaminoglycan chains are added to the core proteins to form proteoglycans. The sulfation of the sugars in proteoglycans and of selected tyrosines and proteins are also occurs in a late Golgi compartment. So there are so many things that occur in the Golgi apparatus. No, so they modify the protein through O-link glycosylation and some glycosyl transferase action in order for it to be specific enough as it is being transported to other cellular compartments. Okay. So the Golgi apparatus modifies the many proteins and lipids, just like what I've said earlier, that it has received from the ER. The Golgi apparatus is a polarized organelle consisting of one of the more stacks of this shape cisternae. So just like what I said earlier, it's made up of at least 3 functionally distinct compartments, termed as the cis, the medial, and the trans. So the cis is the one that faces the rough ER, the medial in the middle, and the trans Golgi, the one that faces or near the cell membrane. So the proteins here, they move in stacks from the cis to the middle to the trans. Okay? So cis to trans direction, this movement may occur by vesicle transport by progressive maturation of the cisternae as they migrate continuously. Okay? Finished new proteins end up in the trans Golgi network, which packages them in transport vesicle and dispatches them to their specific destination, the cell. So, say for example, protein is already formed here. It will be transported here. So, post-translational process will occur, and it will end. The modification will end here. So, the product here will be a mature protein. It will be it will be packed in a vesicle. It will be transported to its proper destination. So say for example, the protein will be transported from the trans Golgi network, the site where the maturity of the proteins produced occur, and it will be transported to lysosomes. But first, let's talk about lysosomes. So what are lysosomes? So lysosomes are specialized for the intracellular digestion of macromolecules. So they contain unique membrane proteins and a wide variety of soluble hydrolytic enzymes, the hydrolases, that operate best at pH 5, which is the internal pH of lysosomes. So an ATP-driven proton pump in the lysosomal membrane maintains this low pH. Newly synthesized lysosomal proteins transported from the lumen of the ER to the Golgi apparatus are carried from the trans Golgi network to endosomes using clathrate-coated transport vesicles, because this is an endocytic pathway. The lysosomal hydrolases contain oligosaccharides that are covalently modified. Uniquely in the cis Golgi, their mannoses are phosphorylated. We know that already. No? These mannose 6-phosphate groups are recognized by an M6P. Okay? M6P receptor protein in the trans Golgi network that segregates the hydrolysis and helps package them into body transport vesicles that deliver their contents to endosomes. So the M6P receptors shuttle back and forth between the trans Golgi network and the endosomes. The low pH in endosomes and the removal of the phosphate from the M6P group cause the lysosomal hydrolases to dissociate from these receptors, making the transport of the hydrolases unidirectional. A separate transport system uses clathrine-coated vesicles to deliver resident lysosomal membrane proteins from the trans Golgi network to the endosomes. Okay, now let us talk about the transport into the cell from the plasma membrane. So we know that the process is called endocytosis, in which localized regions of the plasma membrane invaginate and pinch off to form endocytic vesicles. In most cells, endocytosis will internalize large portion of the plasma membrane every hour. So these cells remain the same size, but because of most of the plasma membrane components that are endocytosed are continually returned to the cell surface by exocytosis. So this is mediated largely by clathrine-coated pits and vesicles, but clathrine-independent endocytic pathways also contribute. Okay. Many of the endocytosed molecules are quickly recycled to the plasma membrane. Others eventually end up in lysosomes where they are degraded. Most of the ligands that are endocytosed with the receptors dissociate from the receptors in the acidic environment of the endosomes and eventually end up in lysosomes. So most of the receptors are recycled via transport vesicles back to the cell surface. So many cell surface signaling receptors become tagged with ubiquitin when activated by binding their extracellular signal. So ubiquitination guides activated receptors into clathrin-coated bits. Okay, they and their ligands are efficiently internalized and delivered to early endosomes. So early endosomes will rapidly mature into late endosomes. So during maturation, patches of the endosomal membrane containing ubiquitinated receptors invagellate and pinch up to form interluminal vesicles. So this is the overall process of how clathrin-coated vesicles are formed during the transfer of proteins from the here as it endocytoses the substances from outside of the cell. So the process, those that we mentioned earlier, is mediated by ESCRT complexes and sequesters the receptors away from the cytosol, which terminates their signaling activity. So late endosomes, they migrate along microtubules toward the interior of the cell where they fuse with one another and with lysosomes to form in the lysosomes where degradation occurs. Okay. Now, how about the transport from the trans-Golgi network to the cell exterior? So this is exocytosis. Okay, so it is almost the same. So from the trans-Golgi networks, the secretory vesicles that contain the proteins for secretions will bud from the trans-Golgi network. Now they will be released there. Synaptic vesicles, which are confined to nerve cells, for example. Now, some endocrine cells, they form from both endocytic vesicles and endosomes, and they are mediate the regulated secretion of small molecule neurotransmitters that the axon terminal or nerve cells needed. Proteins are delivered from the trans-Golgi network to the plasma membrane by the constitutive pathway unless they are diverted into other pathways or retained in the Golgi apparatus. Okay, so this is how the process from the exocytosis from the trans Golgi to the cell exterior. So that ends our lesson. Okay, so in order for you to review the topics that you have discussed, you might want to answer these questions. What is the general flow of proteins and other substances that are transported via vesicles within the cell? How do cells take in and excrete substances? So I hope you will be able to answer those questions. So if you have further questions, you can ask me through my email or your lecturer for the email provided by your lecturer. And you can also key in your questions in the group chat created for your class. Thank you very much. And thank you for being with this class for the whole semester.